say that right. Help me. Okorafor. Thank you. Evelyn Okorafor is going to talk to us about something small that can change something big. Are you ready? Let's give her a hand.
I remember that night. I remember that night 10 years ago when I lost my mom. The very day the doctor certified her healthy and strong enough to go home with us. It was the worst day of my life, and I was very devastated. But in my trauma, I decided to research the reason why people die, even after being certified healthy and strong to go home, or being discharged from the hospital. And that was when it dawned on me that some diseases that you and I have heard about are actually incurable. Instead of being cured, medicine will just have you manage them for the rest of your life. And this is because either the root cause is yet unknown or not fully understood. At that point, I promised myself that I was going to be involved in some kind of drug discovery, whether secondary or primary. And my coming to the US for my PhD was another opportunity for me to explore my passion. Some common diseases that you and I have heard about, like diabetes, cancer, COVID-19, heart diseases, and several others, have been linked to membrane proteins. A class of proteins that exist alongside phospholipids in the cell membrane, which is present in everyone seated here in this room. Now, let's take a quick dive back to our elementary biology, where we were taught that the cell is the central unit of life, and that human beings are made up of trillions of cells in them. And as you can see on the screen, the cell makes up the tissue, which makes up the organ that makes up you, a human being. In case you still don't get it, now, imagine the cell like your house, your beautiful house, that has a wall that shields it from its environment. And imagine that wall as a cell membrane, which shields the cell from its environment. And then, while you picture your house, I know your house has windows and doors that allow you to go out, come in, get groceries, and then shut your door against anyone you don't want. Now, imagine those doors and windows as our membrane proteins, because they act as transporters that helps the passage in and out of the cell of molecules and ions like glucose, calcium, potassium, and others. And then there's a part of the wall that gives it its structure and rigidity. Think about that like our phospholipids. Now, I have a big question for you to think about. What do you think is going to happen to your house if your windows and your doors are distorted? Same question you think about. What happens to the human and the cell if something goes wrong with these membrane proteins? Membrane proteins also act as enzymes, as they help to catalyze several chemical reactions in the body. And this class of proteins are important drug targets. That is why they account for 60% of FDA-approved drugs. But something is wrong again, because just 2% of their structures are represented in the protein data bank, a database where scientists are allowed to go search out the structure and properties of proteins while trying to study them further. And I know you'll be thinking in your head, why? Because these proteins are so important. Why? The reason is because outside of their native environment, it is very challenging to study this class of proteins. Scientists have tried over the years to be able to create an environment that suits these proteins for us to be able to study them. From the onset of using detergents to this era where we use polymers like styrene malic acid, a polymer that has been used to fix the structure of membrane proteins through cryo-EM and NMR. In case you still don't get it, let me explain further how this environment works. Imagine if everyone in this room is given a task to study how a catfish moves or how a catfish behaves. And you will agree with me that a catfish exists in a big water body, right? And we can't all go to that big water body to study that catfish. So what do? To achieve our goal, we have to create an artificial pond or a lake somewhere close by where we have access to our fish, and where our fish will also be comfortable while we study the movement and the behavior of that fish. Now, this new environment we create for our fish is what styrene malic acid and its derivatives or other polymers does for our membrane proteins. My lab, which is Dr. Lorigan's lab, is focused on conducting biophysical and structural studies of these membrane proteins. And my research is basically on improving the application of styrene malic acid, its derivatives, and other polymers, in the sense that we get the right polymer, the right charges, the right size, and the right ratio needed to keep our membrane proteins happy. And because I mentioned earlier that these membrane proteins exist alongside phospholipids, I am also involved in optimizing the composition of phospholipids needed to study our membrane proteins. Because understanding each membrane protein function and structure totally depends on having the right polymer and having the right phospholipids to create a favorable environment for our membrane protein. We got you covered. I have great news for all of us. Because we're not just doing this work for us, we're creating a bigger tool for researchers and drug discoverers, as they have the opportunity to develop more drugs that you and I need to be healthier and to live longer. Because medicine is something universal and is very indispensable. But we can only cure what we truly understand. Thank you.
Let's give Evelyn another hand. We appreciated that talk. So let us know something so small as our protein membranes hold the key to healthy life and quality of living. Our next speaker